Homeostasis is defined as the ability of a body or cell to seek and maintain a condition of equilibrium or stability within its internal environment when dealing with external changes. This means that the bodies or cells are able to keep factors in the body, such as blood glucose, cell pressure, blood pH, within a small range. When these factors are within this range, the body becomes more efficient. For example, maintaining blood glucose in a small range is important, as too much blood glucose can damage proteins in the body, but too little glucose means most organs, most importantly the brain, will be unable to function properly. The core idea behind homeostasis in an organism is the negative feedback loop. Imbalance to the variable of the homeostatic system is detected by the receptor. This is usually the hypothalamus. The receptor recognizes and determines the change in the variable, and this information is sent to the control center. The control center determines what changes need to be made to reach stability and will send a hormone or nerve signal to the effector. The effector is an organ that upon receiving the signal will act to bring equilibrium. Negative feedback comes from the fact that the information about the imbalance causes the system to neutralize the imbalance. It acts to remove the feedback. On the left is an illustrated example of the negative feedback loop. The graph on the right depicts the fluctuation of the variable as it is rising above and below the optimum. The body is only able to keep the variable within a range of values. It is too hard to maintain the optimum. The variable will constantly rise above and below equilibrium as the body experiences and reacts to external changes. Thermoregulation is the homeostatic system that moderates the body temperature of an organism. This is primarily seen in animals. Ectotherms are organisms that rely on external sources to keep maintaining their body temperature. These are also known as cold-blooded animals. Endotherms are organisms that create their own heat as a byproduct of metabolism. Homeotherms, which are a subgroup of endotherms, maintain a much tighter range of temperatures in their body. Maintaining a consistent body temperature is important to the organism. The speed of a chemical reaction in metabolism is determined by the temperature of the reactants. The highest possible temperature will increase the speed and efficiency of metabolism. But temperatures cannot exceed the point where the heat will denature the proteins, as this will destroy the enzymes that catalyze the metabolic reactions, which means the body will not function properly. Low temperatures also reduce the efficiency of metabolism and can cause damage to cells. External temperatures are detected by thermoreceptors in the nervous system, which send this information to the hypothalamus. The hypothalamus itself also has thermoreceptors and will measure the temperature of blood as it passes through and determine the body temperature. The first actions taken by an organism to stabilize temperature are voluntarily. This is behavior that acts to quickly change the temperature of an organism's microclimate. If voluntary action cannot stabilize the body temperature, then non-voluntary actions will kick in. In thermoregulation, the peripheral nervous system and hypothalamus act as the receptor for the temperature change. The hypothalamus is also the primary control center for most of the feedback loops. Animals have many methods to combat temperature imbalance, and so there are several effectors for thermoregulation. The end result is to restore the body temperature of the organism's optimum range. The Carospiny mouse, or Acomys cahirinus, is a small desert rodent that needs thermoregulation to survive. It is found in the hot climate north of the Sahara and can also be found in rocky areas. During the day, the desert heats up to very high temperatures and then cools severely during the night, as there is little in the way of vegetation or clouds to retain the heat. The exact body temperature of the cahirinus is not confirmed, but is known to be between 30 and 40 degrees Celsius. Thermoregulation is important to all animals because of the way substrates and proteins behave as the temperature increases. Due to the variance in temperature of the cahirinus environment, being in the hot desert days and cold desert nights, it is especially important for the cahirinus to be able to maintain its body temperature through thermoregulation. The cahirinus, like many small animals, also has issues to do with high body temperatures due to the excess heat production from metabolism, compared to their small bodies. One of the ways the cahirinus combats temperature change is through changing the size of their blood vessels. In order to cool itself, the cahirinus can dilate its vessels and let hot blood into the surface blood vessels, which will cause heat to radiate from the cahirinus' skin. It can also retain heat when cold by constricting vessels and diverting blood into deeper tissue, which causes less heat to be lost. This can only preserve the existing heat in the cahirinus. It does not create more heat energy. This is the feedback loop for vasoconstriction and vasodilation. If the body temperature of the cahirinus is too high, the temperature will be detected by thermoreceptors in the hypothalamus. The hypothalamus will then stimulate the pituitary gland to signal to the adrenal gland to secrete more epinephrine, or adrenaline, into the bloodstream. The blood vessels then expand, which leads to more heat loss from the skin. The loss in temperature is the negative feedback, and when the body temperature returns to normal, will result in the normalization of the production of epinephrine. If the temperature of the cahirinus is too low, then the temperature change will again be detected by the hypothalamus. The hypothalamus will stimulate the pituitary gland to signal to the adrenal gland, but instead, the production of epinephrine will decrease, resulting in less epinephrine in the bloodstream. This causes the blood vessels to constrict and will cause less heat to be lost. Over time, heat as a byproduct of respiration will increase the body temperature of the cahirinus to the normal. When this happens, negative feedback will result in the normalization of the production of epinephrine. The cahirinus is also able to use the pilomotor reflex to reduce heat loss when cold. Small muscles, known as erector pili muscles at the base of every hair follicle on the cahirinus, can contract, pulling the hairs erect. This traps a layer of warm air close to the skin of the cahirinus and creates a layer of insulation from heat loss through convection. In the pilomotor reflex, low body temperatures are detected by the sympathetic nervous system. The SNS then signals to the adrenal glands to release epinephrine into the erector pili muscles. The APMs then contract, making the hairs erect. The layer of warm air insulates the cahirinus and mitigates the amount of heat loss through convection. Over time, heat as a byproduct of respiration will cause the body temperature of the cahirinus to rise, and the increasing temperature will be detected by the SNS, causing the adrenal glands to stop producing epinephrine. The cahirinus is able to generate heat through non-shivering thermogenesis. This is an ability that is normally limited to newborn or hibernating animals. NST can only increase the body temperature, not decrease it. This occurs in brown fat cells, also known as brown adipose tissue, which is usually found near white fat cells, but can also be found in the brain, liver, and other major organs. NST produces heat by uncoupling ATP from the electron transport during respiration, which causes heat energy to be produced from the combustion of food. In non-shivering thermogenesis, low temperatures are detected by the sympathetic nervous system. The SNS acts as both the receptor and control center in the feedback loop, as the nerves are the organ that determine the low body temperature, but are also responsible for releasing norepinephrine, or Ne, directly into the brown adipose tissue. The norepinephrine binds to the beta-3 adrenergic receptor in the plasma membrane of the brown adipose tissue. Then, through intercellular signaling, this leads to the degeneration of triglycerides in the lipids, which releases the free fatty acids. These fatty acids activate uncoupling protein 1, UCP1, and overcome the inhibition caused by purine nucleotides in the mitochondria. The presence of UCP1 in the mitochondria results in respiration. However, the transfer of electrons in the respiration is uncoupled from the production of ATP due to the fact that the UCP1 increases the permeability of the inner mitochondrial matrix, which allows proteins to pass through and bind to the electrons. This means that heat will be produced from the respiration, but will result in a low rate of ATP production. This heat is the prime product of NST and will cause the body temperature of the cahirinus to rise. The increase in BT will be detected by the SNS. This is the negative feedback, which will stop the secretion of Ne into the brown adipose tissue, stopping NST. The thermoregulatory system of the cahirinus can be disrupted, meaning that it will not function as it should. This can happen because of internal conditions. Mice that live in the far northern parts of Africa experience cooler climates due to the closeness of the Mediterranean. To conserve energy, the cahirinus will lower its body temperature to hypothermic levels, which helps to conserve energy as respiration becomes limited. Only mice that live in those moderate conditions can do this, otherwise the high external temperatures would shock the mouse. This is a well-regulated daily torpor, or state of hibernation. This is a disruption to thermoregulation, as the cahirinus's body temperature stays far below the tolerance range, yet the negative feedback loops will not act to restore the cahirinus's body temperature. The hypothalamus will detect factors such as temperature and humidity to determine whether the climate is appropriate to enter the torpor. The hypothalamus will then inhibit production of thyrotropin-releasing hormone to the pituitary gland. The pituitary gland detects the low levels of TRH and inhibits the production of thyroid-stimulating hormone to the thyroid gland. As a
result, thyroid gland will produce less thyroxine. Thyroxine is a hormone that stimulates many key metabolic processes, the primary example being respiration. Heat is a major byproduct of metabolism, and because less heat is produced from metabolism, the body temperature of the cahirinus will begin to drop rapidly until it reaches hypothermic levels. The hypo will end when the low levels of thyroxine are detected by the hypothalamus, which will increase production of TSH and TRH from the pituitary gland, thereby increasing production of thyroxine and normalizing thyroxine levels in the blood. This will stimulate metabolism in the cahirinus, which will increase its body temperature, ending the topo.